Olfaction, also known as olfactics, is the sense of smell. This sense is mediated by specialized sensory cells of the nasal cavity of vertebrates, which can be considered analogous to sensory cells of the antennae of invertebrates. In humans, olfaction occurs when odorant molecules bind to specific sites on the olfactory receptors. These receptors are used to detect the presence of smell. They come together at the glomerulus, a structure which transmits signals to the olfactory bulb. Many vertebrates, including most mammals and reptiles, have two distinct olfactory systems: the main olfactory system and the accessory olfactory system. For air-breathing animals, the main olfactory system detects volatile chemicals, and the accessory olfactory system detects fluid-phase chemicals. Olfaction, along with taste, is a form of chemoreception. The chemicals themselves that activate the olfactory system, in general at very low concentrations, are called odorants. Although taste and smell are separate sensory systems in land animals, water-dwelling organisms often have one chemical sense. Volatile small molecule odorants, non-volatile proteins, and non-volatile hydrocarbons may all produce olfactory sensations. Some animal species are able to smell carbon dioxide in minute concentrations. Study of olfaction. Early scientific study of olfaction includes the extensive doctoral dissertation of Eleanor Gamble, published in 1898, which compared olfactory to other stimulus modalities, and implied that smell had a lower intensity discrimination. As the Epicurean and atomistic Roman philosopher Lucretius speculated, different odors are attributed to different shapes and sizes of atoms that stimulate the olfactory organ. A modern demonstration of that theory was the cloning of olfactory receptor proteins by Linda B. Buck and Richard Axel, and subsequent pairing of odor molecules to specific receptor proteins. Each odor receptor molecule recognizes only a particular molecular feature or class of odor molecules. Mammals have about 1,000 genes that code for odor reception. Of the genes that code for odor receptors, only a portion are functional. Humans have far fewer active odor receptor genes than other primates and other mammals. In mammals, each olfactory receptor neuron expresses only one functional odor receptor. Odor receptor nerve cells function like a key lock system. If the airborne molecules of a certain chemical can fit into the lock, the nerve cell will respond. There are, at present, a number of competing theories regarding the mechanism of odor coding and perception. According to the shape theory, each receptor detects a feature of the odor molecule. Weak shape theory, known as odotope theory, suggests that different receptors detect only small pieces of molecules, and these minimal inputs are combined to form a larger olfactory perception. An alternative theory, the vibration theory proposed by Luca Turin, posits that odor receptors detect the frequencies of vibrations of odor molecules in the infrared range by quantum tunneling. However, the behavioral predictions of this theory have been called into question. There is no theory yet that explains olfactory perception completely. Main olfactory system. Invertebrates' smells are sensed by olfactory sensory neurons in the olfactory epithelium. The olfactory epithelium is made up of at least 6 morphologically and biochemically different cell types. The proportion of olfactory epithelium compared to respiratory epithelium gives an indication of the animal's olfactory sensitivity. Humans have about 10 square centimeters of olfactory epithelium, whereas some dogs have 170 square centimeters. A dog's olfactory epithelium is also considerably more densely innervated, with 100 times more receptors per square centimeter. Molecules of odorants passing through the superior nasal concha of the nasal passages dissolve in the mucus lining the superior portion of the cavity, and are detected by olfactory receptors on the dendrites of the olfactory sensory neurons. This may occur by diffusion or by the binding of the odorant to odorant binding proteins. The mucus overlying the epithelium contains mucopolysaccharides, salts, enzymes, and antibodies. This mucus acts as a solvent for odor molecules, flows constantly and is replaced approximately every 10 minutes. In insects, smells are sensed by olfactory sensory neurons in the chemosensory sensilla, which are present in insect antennae, palps and tarsa, but also on other parts of the insect body. Odorants penetrate into the cuticle pores of chemosensory sensilla and get in contact with insect odorant-binding proteins or chemosensory proteins before activating the sensory neurons. Receptor neuron. The binding of the ligand to the receptor leads to an action potential in the receptor neuron, via a second messenger pathway, depending on the organism. In mammals, the odorants stimulate adenylate cyclase to synthesize CAMP via a G protein called Golf. CAMP, which is the second messenger here, opens a cyclic nucleotide-gated ion channel, producing an influx of cations into the cell, slightly depolarizing it. California 2+ is then extruded through a sodium-calcium exchanger. A calcium calmodulin complex also acts to inhibit the binding of CAMP to the CAMP-dependent channel, thus contributing to olfactory adaptation. This mechanism of transduction is somewhat unique, in that CAMP works by directly binding to the ion channel rather than through activation of protein kinase A. It is similar to the transduction mechanism for photoreceptors, in which the second messenger cGMP works by directly binding to ion channels, suggesting that maybe one of these receptors was evolutionarily adapted into the other. There are also considerable similarities in the immediate processing of stimuli by lateral inhibition. Averaged activity of the receptor neurons can be measured in several ways. In vertebrates, responses to an odor can be measured by an electro-olfactogram or through calcium imaging of receptor neuron terminals in the olfactory bulb. In insects, one can perform electroantennogram or also calcium imaging within the olfactory bulb. Olfactory bulb projections. Olfactory sensory neurons project axons to the brain within the olfactory nerve. These nerve fibers, lacking myelin sheaths, pass to the olfactory bulb of the brain through perforations in the cribriform plate, which in turn projects olfactory information to the olfactory cortex and other areas. The axons from the olfactory receptors converge in the outer layer of the olfactory bulb within small structures called glomeruli. Mitral cells, located in the inner layer of the olfactory bulb, form synapses with the axons of the sensory neurons within glomeruli and send the information about the odor to other parts of the olfactory system, where multiple signals may be processed to form a synthesized olfactory perception. A large degree of convergence occurs, with 25,000 axons synapsing on 25 or so mitral cells, and with each of these mitral cells projecting to multiple glomeruli. Mitral cells also project to periglomerular cells and granular cells that inhibit the mitral cells surrounding it. Granular cells also mediate inhibition and excitation of mitral cells through pathways from centrifugal fibers and the anterior olfactory nuclei. The mitral cells leave the olfactory bulb in the lateral olfactory tract, which synapses on 5 major regions of the cerebrum: the anterior olfactory nucleus, the olfactory tubercle, the amygdala, the piriform cortex, and the antihynal cortex. The anterior olfactory nucleus projects, via the anterior commissure, to the contralateral olfactory bulb, inhibiting it. The piriform cortex has 2 major divisions with anatomically distinct organizations and functions. The anterior piriform cortex is better associated with determining the chemical structure of the odorant molecules, and whereas the posterior piriform cortex is best known for its strong role in categorizing odors and assessing similarities between odors. The piriform cortex projects to the medial dorsal nucleus of the thalamus, which then projects to the orbitofrontal cortex. The orbitofrontal cortex mediates conscious perception of the odor. The three-layered piriform cortex projects to a number of thalamic and hypothalamic nuclei, the hippocampus and amygdala and the orbitofrontal cortex, but its function is largely unknown. The entorhinal cortex projects to the amygdala and is involved in emotional and autonomic responses to odor. It also projects to the hippocampus and is involved in motivation and memory. Odor information is stored in long-term memory and has strong connections to emotional memory. This is possibly due to the olfactory system's close anatomical ties to the limbic system and hippocampus, areas of the brain that have long been known to be involved in emotion and place memory, respectively. Since any one receptor is responsive to various odorants, and there is a great deal of convergence at the level of the olfactory bulb, it seems strange that human beings are able to distinguish so many different odors. It seems that there must be a highly complex form of processing occurring, however, as it can be shown that, while many neurons in the olfactory bulb are responsive to many different odors, half the neurons in the orbitofrontal cortex are responsive to only one odor, and the rest to only a few. It has been shown through microelectrode studies that each individual odor gives a particular specific spatial map of excitation in the olfactory bulb. It is possible that, through spatial encoding, the brain is able to distinguish specific odors. However, temporal coding must be taken into account. Over time, the spatial maps change, even for one particular odor, and the brain must be able to process these details as well. Inputs from the two nostrils have separate inputs to the brain, with the result that it is possible for humans to experience perceptual rivalry in the olfactory sense akin to that of binocular rivalry when there are two different inputs into the two nostrils. In insects, smells are sensed by sensor located on the antenna and maxillary pulp and first processed by the antennal lobe, and next by the mushroom bodies and lateral horn. Accessory olfactory system. Many animals, including most mammals and reptiles, but not humans, have two distinct and segregated olfactory systems. A main olfactory system, which detects volatile stimuli, and an accessory olfactory system, which detects fluid phase stimuli. Behavioral evidence suggests that these fluid phase stimuli often function as pheromones, although pheromones can also be detected by the main olfactory system. In the accessory olfactory system, stimuli are detected by the vomeronasal organ, located in the vomit between the nose and the mouth. Snakes use it to smell prey, sticking their tongue out and touching it to the organ. Some mammals make a facial expression called phlegmon to direct stimuli to this organ. The sensory receptors of the accessory olfactory system are located in the vomeronasal organ. As in the main olfactory system, the axons of these sensory neurons project from the vomeronasal organ to the accessory olfactory bulb, which in the mouse is located on the dorsal posterior portion of the main olfactory bulb. Unlike in the main olfactory system, the axons that leave the accessory olfactory bulb do not project to the brain's cortex but rather to targets in the amygdala and bed nucleus of the stria terminalis, and from there to the hypothalamus, where they may influence aggressive in mating behavior. Human olfactory system. In female humans, the sense of olfaction is strongest around the time of ovulation, significantly stronger than during other phases of the menstrual cycle and stronger than the sense in males. The Mhc genes are a group of genes present in many animals and important for the immune system in general. Offspring from parents with differing Mhc genes have a stronger immune system. Fish, mice and female humans are able to smell some aspect of the Mhc genes of potential sex partners and prefer partners with Mhc genes different from their own. Humans can detect individuals that are blood-related kin from olfaction. Mothers can identify by body odor their biological children but not their stepchildren. Pre-adolescent children can olfactorily detect their full siblings but not half-siblings or step-siblings, and this might explain incest avoidance and the Westermarck effect. Functional imaging shows that this olfactory kinship detection process involves the frontal temporal junction, the insula, and the dorsomedial prefrontal cortex, but not the primary or secondary olfactory cortices, or the related piriform cortex or orbitofrontal cortex. Olfactory coding and perception. How olfactory information is coded in the brain to allow for proper perception is still being researched, and the process is not completely understood. When an odorant is detected by receptors, the receptors in a sense break the odorant down, and then the brain puts the odorant back together for identification and perception. The odorant binds to receptors which recognize only a specific functional group or feature of the odorant, which is why the chemical nature of the odorant is important. After binding the odorant, the receptor is activated and will send a signal to the glomeruli. Each glomerulus receives signals from multiple receptors that detect similar odorant features. Because multiple receptor types are activated due to the different chemical features of the odorant, multiple glomeruli will be activated as well. All of the signals from the glomeruli will then be sent to the brain, where the combination of glomeruli activation will encode the different chemical features of the odorant. The brain will then essentially put the pieces of the activation pattern back together in order to identify and perceive the odorant. It is a general idea that the layout of brain structures corresponds to physical features of stimuli, and similar analogies have been made in olfaction with concepts such as a layout corresponding to chemical features or perceptual features. While chemotopy is a highly controversial concept, there is more evidence for perceptual information implemented in the spatial dimensions of olfactory networks. Although conventional wisdom and lay literature, based on impressionistic findings in the 1920s, have long presented human olfaction as capable of distinguishing between roughly 10,000 unique odors, recent research has suggested that the average individual is capable of distinguishing between over 1 trillion unique odors. Researchers in the most recent study noted that this estimate is conservative, and that some subjects of their research tests involve testing the psychophysical responses to combinations of over 128 unique odor molecules with combinations composed of up to 30 different component molecules, might be capable of deciphering between 1,000 trillion odorants, while they estimated their worst performer could still distinguish between 80 million scents. Authors of the study concluded, this is far more than previous estimates of distinguishable olfactory stimuli. It demonstrates that the human olfactory system, with its hundreds of different olfactory receptors, far outperforms the other senses in the number of physically different stimuli it can discriminate. However, it has also been noted by the authors that the ability to distinguish between smells is not analogous to being able to consistently identify them, in that subjects were not typically capable of identifying individual odor stimulants from within the odors the researchers had composed from multiple odor molecules. In November 2014, the study was strongly criticized by Caltech scientist Marcus Meister, who wrote that the study's extravagant claims are based on errors of mathematical logic. Genetics of olfaction. Different people smell different odors, and most of these differences are caused by genetic differences. Although odorant receptor genes make up one of the largest genes families in the human genome, only a handful of genes have been linked conclusively to particular smells. For instance, the odorant receptor OR5A1 and its genetic variants are responsible for our ability to smell beta-ionin, a key aroma in foods and beverages. Similarly, the odorant receptor OR2J3 is associated with the ability to detect the grassy smelling odor CIS3HEXEN1-ol. The preference of cilantro has been linked to the olfactory receptor OR6A2. Interactions of olfaction with other senses. Olfaction and taste. Olfaction, taste and trigeminal receptors together contribute to flavor. The human tongue can distinguish only among 5 distinct qualities of taste, while the nose can distinguish among hundreds of substances, even in minute quantities. It is during exhalation that the olfaction contribution to flavor occurs, in contrast to that of proper smell, which occurs during the inhalation phase. The olfactory system is the only human sense that bypasses the thalamus and connects directly to the forebrain. Olfaction and audition. Olfaction and sound information has been shown to converge in the olfactory tubercles of rodents. This neural convergence is proposed to give rise to a percept termed smond. Whereas a flavor results from interactions between smell and taste, a smond may result from interactions between smell and sound. Disorders of olfaction. The following are disorders of olfaction: anosmia, inability to smell; dysosmia, things smell different from memory or expectation; hyperosmia, an abnormally acute sense of smell; hyposmia, decreased ability to smell; olfactory reference syndrome, psychological disorder which causes the patient to imagine he or she has strong body odor; parosmia, things smell worse than they should; phantosmia, hallucinated smell, often unpleasant in nature.